Welcome back to the Business of Biotech, where last week we left you dangling off of a cliff on the edge of your seat with a to-be-continued conversation with current Exsilio CEO and OrbiMed partner and past Moderna CMO, Dr. Tal Zaks. I'm Matt Pillar.
And I'm Anna Rose Welch.
And when you last heard from us, we were testing Dr. Zaks on his philosophy that it's not unmet medical need, but instead curiosity that drives innovation in biotech, and more specifically in the mRNA space. And on that note, if you're tuning in today, but you haven't listened yet to last week's episode, stop what you're doing right now. Listen to last week's episode. Dive right into that one first, lest you find yourself confused, even more so than you usually are, trying to following along with one of my rambling.
Speaking for yourself, Matt.
Interviews. Well, Anna Rose, we definitely want to give them more context of you as well before they listen to this part too.
Just an interloper. Just creep on in here and there.
So let's jump right back into the interrogation. Tal, I was going to ask you, this is where we were going the last time we spoke. I was going to ask you, in this industry where we see so often that money and to some extent regulatory favor reward those who chase unmet needs, and big bonus if those unmet needs are in big market opportunities. How do you go about convincing people instead, per your commentary the last time we spoke, to fund and back curiosity?
Well, Matt and Anna Rose, first of all, thank you for having me back in the program. It's wonderful to be with you again, and I look forward to the conversation. I think the answer is you have to paint a path to value for people. We don't fund curiosity for the sake of curiosity. That's an academic pursuit, and the basic sciences, that is something that we all contribute to as citizens. That's a public health, the NIH is notable in the US for doing that. I think when it comes to a return on investment in sort of the commercial economic sphere in which I live, what you have to do is create a vision whereby you will, if that curiosity pans out, you ask yourself the what if question. What can you do with it that's of value? And show the path that people can actually sign up to say, yeah, we can get it. We realize it's risky. We realize you don't know everything, but if you can do this, here's what it looks like at the end. And the end game is of concrete value to patients out there in the world. And I think that's what garners the investment. We're fortunate, switching gears just for a second, because you asked on regulatory. The U.S. is very unique, and I'm very proud of our regulatory system. Not many people realize this. When you go to FDA and you say, hey, I think this works, they basically tell you, show me the data, and I will do my own analyses. When you go to, outside the U.S., usually people don't have the same depth to do their own analysis. So they ask me, okay, prove it. And then they ask you questions trying to poke holes at it. But the FDA is unique in the depth of the competencies that they have. And I think all of us as consumers should be really proud of it, especially in the US. And it's one of the reasons that most of the rest of the world looks to the US FDA for approvals and as a basis for making judgment. Going back to sort of the curiosity angle, when somebody comes to me with an idea, or back in the day when we were starting this with mRNA, the critical question was not, will it work? Can you make mRNA translate a protein in the human body? The critical question was, if it could work, what would you do with it that would be of value? Mm-hmm. And I think that's where a lot of our energy went in the early days, and that's where a lot of investor sentiment came in back then. Mm-hmm. The company is famous for having raised successive rounds that were, unprecedented at the time because of the potential value that we saw in it. Switching gears today, when we look at investments, we look at things that are cutting edge usually, and the question is always, okay, what is the medicine at the end of the rainbow, and is there a credible path to get there within the financing that you're trying to raise? The company I'm leading now, Exsilio, started with an idea that is based in curiosity. It basically looks at one of these elements in our genome, these genes that can go from place to place in the genome. Think of them as really early primordial viruses. If our concept of a virus is a piece of genetic material that goes to infect somebody else, another genome, well, these primordial viruses or parts of viruses are sort of, we call them jumping genes at a certain stage. They actually evolved to try to jump within our own DNA. And because of that, they have unique ability of where they can go and what they can do, but also evolution has constrained their ability to jump. Otherwise, our genomes would be, junk and we wouldn't be here. Again, for full disclosure, you are now speaking with somebody who believes in evolution. When the company was started, the question was, well, if these indeed occur, and if you map our genome, and we can do that now, have been doing it for several years, almost half of our genome is remnants of various kinds of these genes trying to jump about. Then it's pretty clear that some of them still have that ability to jump a little bit, but evolution has constrained that, otherwise they'd wipe us out. So can we find one of those elements and now, using modern tools of engineering, actually make them better and start to use them as a platform for fixing genes, for inserting a gene where somebody is born with an error where they don't have the gene for that. That's the frequent cause of rare genetic diseases. And that was the idea. And I became excited because the team that came together actually was marrying two avenues of research. One of them is mRNA and lipid nanoparticle. We spoke about that at length. The other one was gene therapy. And we're now living, we already have some gene therapies approved, and we've been using really cumbersome ways of getting these foreign genes, or these fixing genes, if you will, into our genome using what are called viral vectors, so bits and bobs of viruses that are really hard to make and have a whole host of challenges associated with them. And when you can do that, you can only do that once, because the immune system then immediately recognizes it. You can never give that gene again. So you got to get it right on the first shot. And the whole gene therapy industry has evolved to this mantra of a one-time dose, right? Once and done. Well, it turns out that once and done is actually a bug. It's not a feature. We can't do it more than once, even if we needed to. And what this team that formed Exsilio actually came up with, the notion that if we could get this to work, if we could leverage what's already in nature but improve upon it, we can then use mRNA and lipid nanoparticle technology to deliver it. And the beauty is that mRNA and LNP technology is not immunogenic. You can give it with repeat dosing. And so now I don't have to be once and done. I can actually repeat dose and get to the exact gene level I need to get a therapeutic benefit and potentially even a cure. That's what got me excited. And so this is early stage. The team is super curious. They're engineering. They've got these structures up using machine learning, AI, all the usual buzzwords. But the critical piece is, at the end of it, you can paint a path to actually curing somebody of their disease. And that's where you get rewarded.
I think that's a really good point. And that's something too that I've started to hear more of in the mRNA space in particular, right? In a redosable space, we're following in the footsteps of treatments that have been sort of alluring, I think, in terms of that one and done notion, right? Commercially, partially because they are so expensive, right? But at the end of the day, I think that one of the value propositions that hasn't really been seized upon yet by the mRNA space is that ability to redose. And that can really, I think, open up the space to a whole host of advantages, both, especially with patients too, that are probably more familiar with that level of treatment, right? Chronic treatments in the biologic space, for example, it's a risk versus benefit profile, right? That they're more familiar with per se than one and done. But I'm really curious too, in terms of, as you're looking at all of this curiosity, right, in the space and in the mRNA world, how the funding journey has been, right? Have you had to sort of reiterate this story of overcome that sort of allure of the one and done treatment, right, in the gene therapy space as you're working in the mRNA world? And what are you finding is being more rewarded in terms of curiosity in the mRNA space today?
Well, I think that there's been a fundamental shift in funders, investors' willingness to entertain the conversation based on the success of Moderna, Pfizer, BioNTech.
Yeah.
Because what that success has demonstrated is that it is doable to give a human being mRNA and an LNP and go make a protein that will have a therapeutic benefit. And you can do it at scale, and it is safe. And so a lot of things that would have probably taken us, a decade or two to get to, were actually accelerated to the benefit of this technology by the utilization of these vaccines. I think where you see the conversation today, many companies and many investors are looking to invest in sort of next gen, okay, what can we do with this technology? And of course, the space that I'm in, and there are competing companies like CRISPR, Beam, et cetera, that are also trying similar approaches, trying to get away from traditional viral vectors for gene therapy, but actually leverage what the capabilities that have been demonstrated mRNA and LNP can do and bring them into the gene therapy world. It's going to take some time, but between that and the new technologies that are emerging, I'm very optimistic that we will see new medicines come about that leverage sort of both streams coming together.
Are you
just to say one last thing, I think it's long been recognized that actually progress is not really the Archimedes in the bathtub moment of aha, but it's more like people taking things that have matured to a certain point from different fields and bringing them together. And that's where you get sort of true innovation. One example for me that I'm super always happy to discuss and proud of is the journey of the cancer vaccines. I started my career as a postdoc working on that eons ago. When I joined Moderna, I sort of realized that between what we now understood about cancer and what the immune system recognizes, between our ability to do sequencing that we never had before of every person's tumor, and our ability now to use mRNA technology to create a very particular type of vaccine that stimulates just the right part of the immune system to go after cancer, we should be able to put together a personalized cancer vaccine. And we did. We started it back towards the end of 2015 when I was at Moderna, and the results of a randomized controlled trial have recently shown that it looks like that personalized vaccine can actually prevent recurrence of melanoma almost half the time if given in the right setting. So it's these strands that all come together. For Exsilio, it is the understanding of sequencing evolutions, these jumping genes, and what you can do with modern protein engineering tools, and then what you can bring from mRNA and lipid nanoparticle. And everybody talks about AI and machine learning as the next enabler, and we're starting to see how that's accelerating our ability to do discovery in almost every place you look as well. So it's these pulling on different strands where curiosity has given us new tools, and how you put them together to actually create new medicines. That's what's fundable.
Have you found that, from an education standpoint, when you're talking to investors and working with the investor community, that there has been any sort of education that's been needed to sort of help ease that transition from, we know what mRNA LNPs can do in a vaccination context, right, to now here we are exploring therapeutic cancer vaccines. We're looking into protein replacement, gene editing with mRNA and LNPs predominantly. Have you found, right, that narrative, that education, that awareness is needed in certain areas more so than others, or are people pretty up to speed?
I think there's been a dramatic shift in terms of people understanding of the basic technology. Now, that being said, And investors, by and large, especially the biotech ones who are in this space, they're a pretty savvy bunch. But education is you can never do too much of it. And the fascinating thing about it, for me at least, and it goes back to my root as a physician, the biggest challenge every physician has, and the one that we most often fail at, is our ability or the need we have to explain similar pathophysiology processes of what the disease is to different people in different language coming at us from a different viewpoint. And everybody has the experiment of going to a physician and not understanding, well, what is it that they are actually saying? And trust me, the physician understood what they were saying, but the problem is they didn't figure out how to relate it to the patient. And it's on us to learn how to explain things in different language. And it's true in pharma as well. You have people with different expertises. You have the engineering folks, the research folks, the clinical folks, even the finance folks, and they all kind of have their own language. And the fascinating thing is, it turns out it's actually inbred into into our culture. When I was a medical student, I read a fascinating book. There's a Canadian philosopher called John Ralston Saul, and he wrote a book called Voltaire's Bastard, and the subtitle is The Tyranny of Reason in Western Civilization. And basically, he sort of claims that, reason doesn't lead you necessarily to morally correct choices, which is fair. And one of the subplots there that caught my attention is this notion that ever since the Middle Ages, when we started to specialize, every specialty started to sort of enshroud their knowledge in their own language. And that was a barrier to other people because your power was your knowledge. And so you wouldn't share it except with, the cognoscenti of your trade. And so you started to speak in lingo that nobody else could perceive. And boy, do we still have it. By the way, physicians are the absolute worst offenders to this. And I'll give you a simple example. A patient can come to the office and their platelet counts are low. I have no idea why, but because they're low, they will have a certain type of skin rash. And the patient sits there, and the next thing you do is you tell them, well, sir, you've got idiopathic thrombocytopenic purpura. Well, I just told them that you've got a low platelet skin rash for reasons that I don't know, except I said it in Latin. Idiopathic, I don't know. Purpura, skin rash. Thrombocytopenia, your platelets are low, right? And so every profession sort of has that. And the fun for me has been to sort of try to decipher what the other guys actually mean when they use that, and that's within pharma. But when you're speaking about what it is you do, my PhD mentor, the late Shraga Segal, pulled me aside once and said, Tal, if you can't explain what you're doing to somebody in kindergarten, you don't really understand what it is you're doing, right? And so I think on all of us, it's incumbent, those of us who are fortunate enough to be doing the cool stuff that we are, we have to be able to translate that to everybody, to a language that everybody can understand. Otherwise, what we do will be for naught. And that's frankly where you guys come in as well. And I'm so happy to be here because for me, it's an opportunity to share with you and with the rest of the public what's so cool about the stuff that we do.